Hello everyone, welcome to ITLS Academy. Myself, Advocate Priyanka. Today we are going to talk about the introduction to biostatics. So in continuation of our topic, that is the clinical research and regulatory affairs, we have reached to lecture 17, that is introduction to biostatics. So as I always request that please make it interactive session. You can post your comment, you can post your queries after watching the video. For this topic today I have added some content. So first we will go through that what highlights of previous session as usual. Then we will move to our today's topic, that is introduction to biostatics. So in that first we will see that what is biostatics, then what are basic techniques of statics, then application of biostatics, then types of biostatics, then randomization, hypothesis testing, and sample size estimation. Basically, these are the three core principles for the biostatistics. Now, we will move to first like in with previous video what we have talked. So in previous video, we have talked about the clinical research and the media. Media here is basically social media. So we have talked that how social media or digital media is affecting the clinical research. Is it beneficial or is it harmful? So first question is that why it is required to use social media. Obviously it is required to advertise, because for the clinical research, for the clinical trials, we need to reach more population, more people, and it can be done through the advertisement also, through the advertisement only. So basically advertisement is two types: digital media and conventional method. Conventional method, we go through that also, but if we use only conventional method, then we will not be able to, or researchers will be not able to get the more participants. That's why nowadays they use the digital media. So then we have talked about that what is the use of social media in the field of clinical research. As I said just that it is for the recruitment of participant of clinical trials. It is to target diverse population, because nowadays everyone has mobile, everyone has internet. So by using the digital media, by using the social media, we can target different populations from the you can say from the interior side of the country to the from the developed side of the country. Whole population we can target. Then next, making research to more patient centric. After that, conduct of virtual and teledermatology clinical trials. So we have talked that what is the teledermatology clinical trials. It is basically for related to skin. If you want to know in the details, you can go and see the previous video. Next, we have talked about that how to use of social media to reach wider audience. So it's not like that if you are using social media and you will get the more and more audience. For that you will have to follow few steps. There are few techniques so you can reach a wider audience. First step is that for the posting anything, first you will have to identify that what is your goal, what you want to I mean which type of audience you want to target. Then second is identify target audience, like match it to a social media platform. Like if you have identified your goal, then you have targeted your audience, then you will have to match the social media that your audience are using which platform. In that platform you will have to do more focus. After that you will have to see like that the others from the same field for which you are going to post, what they have posted, what they have done. For that you can go and visit the various lab websites who are working related to you can go and see the other researchers posts related to you. After that you have to prepare the novel and engaging content. And it's not like that only you have to prepare it, you have to share the content also and track the responses, that what are the responses, track the comments, and accordingly you have to reply. And then only you will be able to get the more benefit of the social media. I mean researchers will be able to get the more benefits of the social media. Then we have also gone through that what are the challenges of use of social media in clinical research. Like obviously first challenge is that privacy and data safety. So there are few other challenges also. You can watch in the previous video, that is the lecture 16. Now, without wasting the time, we will move to today's topic, that is the introduction to biostatics. So, biostatics has two words, bio and statistics. So, what is the bio? As I know that you all know that you all must be knowing that what is the bio. Bio basically refers to living organisms. So, like biology, a study of living organisms. So, living organism is basically include included in the bioterminology, like plants, animals, microorganisms, all will cover under the bioterminology. Then what is the statistics? Static, Statistics is basically the collection, description and analysis of data. It is like you have to collect the data, you have to observe the data, you have to analyze the data. All these things are done under the branch of study of the statistics. After that, we will see that what is the basically statistics. As I told you that it is basically to the collect, observe and analysis of data. So in this, what is the data? Data is basically the information collected through observation or experimentation. Like blood pressures, patient gender, height, or different other things. So you can say that all these things will come under the data. Now, what is the next? Next is variables. Variables is like that some characteristics that can change or take on different values. Like if we talk about the age is changing. It is a continuously it is changing. So that can be the variable. Then disease stage, if someone has any disease like some cancer or some other disease, it will increase in the different stages. So it also changes, it can be also variable. Or treatment type, like in starting there you are giving the some different type, I mean doctor, and then in next they are giving the some different treatment types. So these all things are variables. Next, descriptive statistics. It is basically method to summarize and organize data. Like researchers have a big group of data, so many persons, so many symptoms of that person's, other parameters, like you can say other variables. So in this they summarize and all organized all this data in a one way with the some there are some techniques, or you can say some process like mean, medians, through they calculate the all data in a group of patients. And like for example, if you take that measure of central tendency, like calculating the average blood pressure reduction in group of patients after administering a new medication and determining the median recovery time for patients after a specific surgery. So this is basically called the descriptive statistics. Now, probability. Like probability is like it is not now, but it can be occurred. So you can say the likelihood of an event occurring. For example, a risk factor is associated with an increased or decreased likelihood of disease. Like say if someone has some problem, and we say no that okay, if he has this disease, like if he has diabetes, he has risk of paralysis attack if he will not take care of. So that means that paralysis attack is not now, but it can be go through. It can be happen in the near future or in the later future, or it cannot be also happen. So this is the basically risk factor. It is not like it will happen. It can be happen or it cannot be happen. So this is called the probability. All these things are used in the statistics. Now we will talk about that what is the biostatistics. As its name is only saying that use of statistics in the bio is called biostatistics. So application of statistics in biology, how it is used? It is used to design studies, analyze results and draw conclusions in field like medicine, public health, genetics and ecology. It is done by collecting data from experiments, from hospital, through surveys, through clinical trials. It is helpful for researchers to work on new treatment method, analyze disease pattern. So why biostatistics is required, or why it is useful? It is useful because if researchers have a large set of data, how will they organize it? How will they make a conclusion? So for all these things, biostatistics is a very important tool. Now, what are the types of biostatics? First is descriptive biostatics. We already discussed in previous slide. Like it involves summarizing and describing feature of data set. Like there are so many symptoms, so many persons, all have so different, so many you can say characteristics. So to summarize all data comes under the descriptive biostatics, involves summarizing and descriptive feature of a data set. Common measures or common methods include mean, median, mode, standard deviation, and frequency distribution. If you want to know more about these things, we can take a different lecture for this. Then used in reporting disease prevalence, demographic statistics, and hospital records. To maintain the hospital records, we use this descriptive biostatics. Then next is inferential biostatics. Make prediction or inferences about a population based on a sample. So in first, what they are doing, they are summarizing the data. In second, they are not only summarizing, they are making their prediction, they are inferencing the data. Like they have the different set of data with the samples. So basically they will make the prediction in this type. Techniques include like hypothesis testing, confidence intervals, regression models, and ANOVA. It is essential in clinical trials and epidemiological studies. So it is not like that if anyone will only summarize the data, it is much required. But after that, in next step, obviously when we have to do prediction when we are summarizing the data. So for that, this is used. Next is analytical biostatics. Analytical biostatistics is basically focus on identifying association or causal relationship between variables. Like what we talked, what is variable, which is the changing factor, like age, like disease stage, blood pressures. All these are variables. So in this analysis, in this type, basically they see that what are the correlation between the different variables. Is there any causal relationship? Is it like that if age increasing, BP is decreasing? I'm just telling this for the example. It is not like that. If age is decrease, if age is less, BP is high. So is there any correlation? So for these things, this type is used. It is used extensively in public health to explore risk factors and disease determinants. Now last one is clinical biostatistics. It basically applies a statistical method to a clinical research, especially trials and treatment efficacy studies. So clinical biostat, only its name is suggesting that it is used for the clinical research, clinical trials. Others are also used for the clinical research trials, but it is used for to the analysis of result for the clinical trials. It involves survival analysis, meta-analysis, and Bayesian statistics. So for these details, we can talk later. Just for your curiosity, I am explaining this one like Bayesian statistics. This so this is what is this? This is basically like, see, it is basically like if you have any belief from the previous, and if you have now some known update that why this belief was. So to make updated belief, this is basically used. So like we will take a general example. Like, see, if anyone nowadays, what our parents say, that we should not cut our nails in the night. It is their belief. Now we have I mean time is changing, so now we have understood, because earlier in night there was no light, there was nothing, so that's of because of that it was believed that we should not cut the nails in the night, because if there will be the dark, it can more cut, it can be harmful. So to make sure that it was the earlier belief, but now we know that why was the belief. So now we have the updated belief. Even our parents have the updated beliefs that we should not cut nails in the night. It is not like unauspicious, it is because for the our safety. But nowadays there is light, everything, so in that way we can cut our nails in the light. So this method, or you can say this principle, is used in the for the clinical research or clinical trials through the Bayesian statistics. For detail, we can take on separate class, because it is all to explain all these things is not possible in this introduction video. Next is what are the application of biostatics? So application of biostatics are in different fields. First is like public health. It helps in monitoring disease outbreaks such as COVID-19. Like when COVID-19 happened, how did you know that how much data, how percent people are in suffering? So for all these to summarize data, our researchers use the biostatistics to support vaccination strategy, health surveillance. It also influences policy decision regarding healthcare budgets. So biostatics is not like only for the analysis of, you can say, analysis of medical things, symptoms, analysis of disease. It is also for the how you are going to manage your budget for the some clinical trials. For that, we also use the biostatics, or you can say we use statistics. Now, second is medical research and clinical trials. It medical research and clinical trials. It is used in drug development by evaluating efficacy and safety. It is used to calculate sample size, analyze treatment effects, and monitor side effects. Third is genetics. It is helps in genetics. It helps in genetics also. It helps in interpreting gene expression, like what are the gene expression for a different age group, and accordingly they summarize the data, and accordingly they analyze the result. It is also used for the psychology and behavioral science, like what are the to quantify cognitive and emotional response, like that how men have the emotional response, how women have the emotional response, and accordingly they summarize the data and they give the some you can say some mm, result, or some views that okay, in man these things can happen, in a woman these things can happen. Measures effectiveness of therapy, that basically affect the therapies, like psychology therapies when it is given. So it is helpful for that. So these are the types of biostatics. Now we will see that basically what are the core concept of biostatics. So they are basically the three core concept of biostatics: randomization, hypothesis testing, and last one is sample size estimation. So first we will see that what is the randomization. Randomization is as its name only. I hope you, I know that you most of will just understood by listening the name, that randomization. Randomization is basically to choose, it's not like to choose a one or two, it's to choose a group of people, or to choose a group of things from the uh, from a large population. So it is basically one of the oldest and most important idea in statistics, playing several role in experimental design and inferences. It is process to assign study participants to treatment or control group by chance. Like see, when we play the game, in that some games we keep the ball, and in that we keep the letters, we keep the chits, and then we say that like if you will tell anyone that you do this, but they will tell that it's a biasness, why will I do this? Then we make some chits and put in the ball, and then we tell others to go and pick one chit, and accordingly it is decided that what work they will do. So then no one can say that it is biasness. Second is, it is required to make balance while selecting participants. So if we talk about the clinical trials, so in that also it is used, because it is used to make balance while selecting participants. It is not like that you can select, if you have a group of population, on which basis you will select. Like if you have generalized on the basis of age, basis of symptoms also. So and after that you have to select few. So in that they do this randomization process. So that's why it will be there will be balance when selecting the participants for the clinical trial. It will be like some unknown factors also include, some known factor also include. So this is the, you can say, basic or oldest core concept of the biostatics. Next is hypothesis testing. Hypothesis testing is basically a structured method to determine if the finding of a study provides evidence to support a specific theory relevant to a large population. So for this, we put our assumption about the population and then we do this test. So for this test, there are 4 steps. First is defining the hypothesis. We will have to first define our hypothesis, that for the particular research, what hypothesis you want to put. We will put one hypothesis, then we will develop a plan for analysis. After that, we will example the sample data, and then we will interpret the final results. Now, defining hypothesis, like there are 2 types of hypothesis in general that it is defined. Like you will put one hypothesis, and for that you will categorize in the 2 different. You will put one. You will put your one hypothesis like about this population. You think that, and then we will divide it into 2: null and alternative. Null, the default assumption, usually stating that there is no effect or no difference. Like what you are saying, it is not right. It is, there is no effect or no difference for the age group, for the genders. An alternative hypothesis is like that the statement the researcher is trying to prove, suggesting there is a significant effect of difference. That means what you are suggesting, what you are trying to prove, it is there is some difference. After that, what we do, what the researchers to develop a plan for analysis. So they just put a significant level, they set the significant level, and researchers usually set a 0.05, that is 5% significant level, p value, that's called p value. That 5% setting means like 95% population have no such effect, but 5% population has such effect. So they define that significant level. After that, they examine the sample data. A random sample is collected from the population, what we talked in the randomization, and then data is analyzed using an appropriate test like t-test, z-test, to produce the test statistics and a p-value. After that, they get the p-value and then they draw a conclusion. So how they draw conclusion, like if the p-value is smaller than significant value, that means what significant value they have put, like 0.05. So if it is less than that, that means result is considered statistically significant. There is something, there is what we have, researchers have added the hypothesis, there is something. If the p-value is greater than significant, you fail to reject the null hypothesis, that there is no difference. So for this, we can see through a example, like the first researchers put a hypothesis that they want to test a new drug effectiveness in particular sex, like female. So they basically, what they have a hypothesis that a pharmaceutical company wants to test if a new drug significantly lowers blood pressure in female patients more than male patients. They want to test this. For that, they have put 2 hypotheses, like first null, no difference. Second alternative, like yes, there is the difference. Then what they do, they will conduct a clinical trial. They will collect the blood pressure data from sample of for the both male and female. Then a test will be performed, such as t-test. After that, when t-test is performed, after that result will come, and then they will do the result analysis. If the p-value calculated, like the p-value calculated comes 0.02, which is less than 0.05, then researcher would reject null hypothesis. They tell what they were telling, that this medicine is going to affect differently the both male and female. It is the right, it has some significant effect, some significant difference. But if anyhow it comes like more than 0.05, then they say no, it is there, it is a null hypothesis. So this is the hypothesis testing and how it is done. Now last is sample size estimation. So also, like, see, if clinical trial is done, how it is defined, that how much sample will be there. It can be very, for some we need very large sample, large population. For some we need a small sample. So it is act of choosing the number of observation or replicates to include in a statistical sample. Sample size is important feature of any empirical study, in which the goal is to make inferences about a population from a sample. There are four ways to define the sample size. First is use of formula. There are many formulas by which we can use the sample. You can define the sample size. Then there are ready-made tables also. By looking in that tables, you can define that, okay, for your thing, how much sample is required. Then there is nomograms. There are some computer software programs like PSS, Unquery, and free options like GPower, OpenEPI. So there are so many computer soft programs. So biostatistics is not a very simple thing. It's not a very simple study. It is a very vast study. It has different features, different branches. So to know all the details, we will have go through all the tests, all the softwares. We will have to see all the things. So today I gave you basically the introduction to biostatistics. So you can understand that if you will choose the biostatistics, or if anyone asks you something in interview that do you know biostatistics, so at least you can tell that yes, I know, and you can explain them that what it is, what is the use of it, what are the applications of it, what are the main core concepts of this. So we have talked a lot about biostatics introduction today. Now if you have any queries or any doubt in this session or in previous session, as I repeat always, so if you have any doubt, please write in the comment section. You can reach me to me through that, and obviously I will come with a doubt clearing session. And next topic is going to be design and conduct of observational studies and clinical trials. So for these clinical trials, we have talked a lot, but what is the observational studies? And basically in this video, in next video, we will see that what is the basic difference when we are designing and conducting observational studies and clinical trials. Is there any difference or there is not difference? So we will see that in that our next topic. And as always, I acknowledge that images are from the Google. Some materials are also from the Google, from the free study sources. So thank you so much to you and to Google also. And please don't forget, follow ITLS Academy on Instagram, Facebook, WhatsApp, LinkedIn. You can get in on the Google Play also. If you want to be in touch, if you want to see next videos of mine or to others also through the ITLS Academy, don't forget to subscribe our YouTube channel. Thank you so much for watching. Like, share, and subscribe. Don't forget it. And stay tuned. I'm coming with the next topic in next video very soon. Thank you so much. Till now, till then, bye bye.